One of the other projects that was funded in our forestry and environment competition, the Genome Canada competition, was the project that's entitled Applied Metagenomics of the Watershed Microbiome. And this is a project that's being led by Judy, Renton, and Patrick Tang, and a large group of people from BC CDC as well as the Genome Sciences Center and so on. So we're delighted that Judy is going to join us and Patrick is going to join us for the panel then also. So please join me in welcoming Judy Renton.
Thanks, Brad, and it's my pleasure to come here and represent a multidisciplinary team. And I think I have a particular perspective because for most of my working day, I work in the Ministry of Health. I am a professor of medical microbiology and interested in infectious diseases, but I run a large laboratory, which is a health service laboratory. Well, why are we looking at Watershed? Well, hopefully I'll explain that. And I wanted to take a particular tack after Brad invited me to speak. I thought what I would be able to share with you is some of the challenges from the partnership perspective. Because in fact, I wear two hats. I'm not only a scientist, I'm actually a client of this project. Being a member of the Ministry of Health, which takes the lead for safe water and all of the implications of safe water in British Columbia, and because we have a legislated mandate to monitor and carry out surveillance, we're very interested as a ministry in looking at better ways of actually doing this, and hopefully I'll be able to share that perspective with you. And as Brad said, Patrick Tang, who's project co-lead, is very much the scientific person who's going to join me on the panel. And Dr. Natalie Prestojecki is also here, and she's our lead as an environmental scientist. And if there's any questions I'm going to ask that Natalie can also address more of the fundamental and basic science from the ecological or watershed perspective. We work very well as a team together, and if I have a feeling today, it's because I'm not acknowledging perhaps some of the details of the work that's being done up to date, and some of the partners that are not represented here today. But the team is forging ahead. We're now about 16 months into the project and looking well. But back to the problem and back to the perspective that I said I'm going to take. The challenge that we're addressing is that of water. And as this quote says, it may become, in fact, an issue that oil was previously. You'll all be familiar with some of the aspects of water from a health perspective. And in fact, we are going to focus in this, and are focusing in this project on surface water. It's possible that we may extend some of these very exciting tools that we're discovering into groundwater, but our current focus is on surface water, and not quantity, but quality, and quality from a microbial perspective. But this more up-to-date quote from 2013 notes that although, and there's some debate about how much of the world's freshwater Canada actually claims to have, we nevertheless are in an enviable position, and sometimes that position can be a challenge. And of course it goes without saying that water is a very precious resource. In fact, some people say, as this author did, water is life itself. And then she goes on to the challenge that this team is taking on, and that is not only Canada, but many people across the world think that water is limitless, and we often waste and pollute it. Back to then the role of the Ministry of Health and other ministries that I'll talk to you about, the regulators and the policymakers, you will be reminded of the notorious, if not infamous, tragedy of Walkerton, Ontario, which is still being written up in the health literature as to its impacts on the people that survived and those that passed away in that contamination event. But I would also like to point out from an ecological perspective, which is the perspective, as you'll see, that this team is taking, that the impacts of pollution and the destruction that's happening in terms of the care of our water impacts not only us humans, animals, fish, and the ecology, the environment. So we waste and pollute it at our peril. So our challenge, the challenge that this team has taken on, is again very similar to some other projects today, is a challenge of developing better ways to monitor the quality from a microbiological perspective of our surface water. And I would like to start by pointing out from the perspective of a client need that anybody that deals with drinking water works within a fundamental paradigm called the source-to-tap model. If you worked within health and you have a very narrow focus around human health, you would be then thinking only of monitoring and looking at tests at the tap or at the point of consuming. And what, in fact, people from a water management perspective are doing is using a model that is holistic, that is in fact what we would call in healthcare system quality management system, where we work together in a plan, do, check, act cycle upstream and in the middle and then downstream as the water flows from its source down to its use. And so that model or that paradigm is called source to tap. And so our challenge is a focus on the source. That is a very needy area where less attention has been paid than monitoring strictly for the narrow focus of human health, more upstream in that source-to-trap model and into the watershed. Now, a word or two about more details of the challenge, very similar to the previous speaker, Nicholas. Our approaches are nearly 100 years old. They're culture-based. They're very slow, and although they do have an advantage, many people don't understand the approach that's currently used. This is obviously a petri dish in a water monitoring lab looking at contamination events happening as reflected by these colonies growing on the petri dish. These tests, nevertheless, are very slow. They're quite inaccurate, and that is because they take a risk-based approach. So many decades ago, it was discovered that when there was fecal pollution of our water, that this event would be able to be reflected in the presence on a petri dish after an overnight incubation of indicators of fecal pollution. And so the approach that has been taken, as I said, for nearly 100 years has been bacterial indicator testing, again, more at the tap and less holistically. And so, given the fact that now that we know using these recent tools of genomics that less than 1% of bacteria, and that's all that this captures, are actually cultivated, you can see that our understanding and our knowledge of the dynamics of contamination events is relatively limited. I should point out that another infamous outbreak that happened in Canada was due to a parasite called Cryptosporidium, and we had many beaver fever outbreaks here in British Columbia. Those organisms are transmitted also through drinking water, as are viruses, but we have no idea and we have no tests that are really practical, real-time and accurate that are currently available. So this is our opportunity and our challenge to move us from a decade-old culture-based monitoring that's looking at a holistic approach of the water system, to the water system, into the real-time opportunity that molecular assays will have. And the intent of this program is a discovery program in which we will determine some biomarkers that will indicate fecal pollution, but will be applied to a format which you're all familiar with, which would be molecular assays like a real-time PCR test, not only to monitor, perhaps as one of the other projects noted, a high-throughput program for monitoring microbial pollution, not only of bacteria but viruses and parasites, but also that we would be able to find markers of pollution, microbial pollution, that point to the source in the watershed of that pollution, and thereby integrate the management cycle and prevention cycle in the watershed to prevent pollution downstream. We would be able to attribute the source through another PCR test opportunity. Oops. The challenge is not only the science and the work on transitioning from culture to molecular assays, it's actually the challenge of, as everybody is facing, I believe, of all the projects that I've heard today in this room are facing, is the transition, the translation into use. But I should pause for a minute and explain a little bit about our study design. We are, as I said, about 16 months in, and we are working with, in that paradigm of source to tap with our watershed managers, policymakers in government at the provincial and federal level, clients of many sorts, including laboratories like our own, to partner in 3 watersheds. Each one is representative of a certain impacted type of watershed. We're working in one watershed that has a very well-known impact from human pollution, human sewage pollution. Second watershed that we're working in is a well-known impact from agricultural activities, and the third is what we call a protected watershed. While there is some natural activity from wild animals, there's no animal and human activities allowed in that watershed, and that's our third watershed. We've been sampling regularly we're nearly complete the sampling processing phase, collecting all the metadata that will be analyzed. The samples are then processed for all groups of organisms, which is no mean feat. We've been developing methods as we move along, and we're looking for not only viruses, the smallest group of microorganisms, and bacteria in the middle. We're also looking for eukaryotes, and process them in a way that will focus on each of those groups. And then those samples have been prepped, and we're now they've gone through 2 approaches in terms of their processing, both an amplicon-based and a shotgun-based, and the data is beginning to flow in. So we're moving into, at about the halfway point, some exciting data analysis that will point to the biomarkers. But I think that whole group of activities, which has been fantastic and which publications are already starting to flow out of that activity, is probably, in my opinion, remember I'm a client as well, probably one of the lesser challenges to meet the fact that the biomarkers are going to be actually picked up by the clients in the field. That is, people like the Ministry of Health or the Public Health Agency of Canada or the US EPA. So how do we transition from culture to molecular biology? How do we go from, maybe not a DNA sequencing approach, although maybe it's going to be PCR, do we replace cultures? What are the benefits? What are the costs? Is it going to meet the needs of the client? For example, PCR will not tell you if an organism is alive or dead. How are we going to do that? We'll have those kind of challenges, but we are moving in that direction. So once again, the current microbial monitoring challenge is, let's look at the source to tap model, not one end and only one constrained piece, and it is a complex environment with many stakeholders. At this point, I'd like to acknowledge the GELS team of a team that works across Canada. We have legal experts from McGill, and we have people in British Columbia that are doing interviews and have completed surveys and are already hearing back from the stakeholders with many observations on the fact that water governance in Canada is very fragmented. We're looking at how we manage water here in Canada and comparing it to the United States. They have some huge advantages in being somewhat centralized around the US EPA, as opposed to the fact that compared to the fact that here in Canada, health and water are provincial responsibilities. The overarching federal responsibility is one of just guiding and producing best practices and guidelines, but each of the provinces has its ability to either ignore those guidelines or follow them. So we have a very fragmented and therefore even more complex challenge than someone working in the United States in terms of translating these tests to the public. And then again, looking at ecological health as opposed to just human health, but we won't forget that human health is also a challenge. So while we will be producing a PCR format probably that will replace a culture-based monitoring environment, and we will continue to take a risk assessment approach that will indicate fecal contamination that will replace a slow and inaccurate microbiome monitoring test. The format has to be acceptable, and have we gone there as yet? I mean, we've heard from our clients who are interviews that they want something cheap, they want something fast, they want something accurate. This has to stand up in court. This is a test that the government will build into our regulations to replace the old monitoring using culture, which is in our law. Basically, if you look at the Drinking Water Act for British Columbia, in our jurisdiction, it says that you must have no more E. coli there, you have not allowed to have anything, and it's specifically stated there. We need to replace that regulation to enable these kind of faster, better, but cheaper tests. So in summary, what we're anticipating to produce, and we're looking for biomarkers, is not a metagenomics test itself. We're going to be producing a format, because real-time PCR in our lab, we've gone from 7 PCR tests in the last 5 years up to 47. That's from a clinical perspective. The environmental world is following, albeit slowly, into that world of molecular microbiology. So we are going to be developing, anticipating a panel of PCR biomarkers, and we're going to be therefore basing our metagenomics test on the information that we gain from the science. So a better method to detect water pollution earlier, a better method to detect pollution upstream before consumers of any sort are taking that water in. So what happens now in my laboratories, we have culture-based methods. They're incubated in the traditional way. 18 hours later, we know the result that there's E. coli there. We don't know if there's parasites there. Meanwhile, the people and the animals and other people have been consuming that water for that period of time. So obviously, detecting pollution upstream earlier is important, and for correcting the mismanagement in the watershed that allowed that pollution is also important. And so, again, developing 2 test kits, both to monitor it in a high throughput, faster way, and to attribute where that pollution has come from is our anticipation. And the next 2 slides is just a summary that Patrick put together, hypothesizing what some of the profiles will be. We may not focus, we're certainly not going to just focus on pathogens of animals or humans. It may be that as we look for biomarkers, we're going to be looking for patterns and profiles that are consistent with healthy water versus fecally polluted water. And for example, if we were monitoring the quality of the water and we had a healthy watershed pattern, and then we saw signals by changes in the patterns that were coming up in this PCR test in the panel, that's what we would declare as an indication for action. And then it's entirely possible that depending, right now there's a science called microbial source tracking, and it's very inaccurate, and it's still not into the molecular world and will not stand up in court. But it's trying to demonstrate that pollution comes from humans, or does pollution that's in your water come from agricultural activities? Where does it come from? And again, the biomarkers with the microbes that we're looking at will be attributing source so there can be corrections upstream in the watershed. So in conclusion, I'm giving less of a scientific report, although the project is moving ahead extremely well, and if you're interested, Patrick can tell you a little bit more about the methodologies and the work that's going on. We're now in the process of receiving a lot of data and into the bioinformatics stage. It is a complex challenge, and we don't anticipate that the next stage will be any easier than the groundbreaking that we did in the first phase. In fact, as I said at the beginning, again, putting on my hat as a client and knowing the challenges of the ministry requiring very solid, accurate tests, we can validate that test. We'll find the biomarkers. We'll probably be able to validate the test. The challenge will be translating that scientific information into a usable product that will be taken up by people like the people that I'm accountable to, that develop the regulations, the people that wrote that Drinking Water Act that talk about petri dishes. We will have to translate what we're finding in science into that regulation, and that will drive, at least in our jurisdiction and in other jurisdictions, the changes that we hope to impact by taking on this challenge. So with that, I'll conclude. And again, if you have more questions that you're interested in terms of methods, then please do, during the panel, please ask them. Thank you.